Now we will discuss about biochemistry, metabolism. First we will discuss about enzyme terminology. An enzyme's name often describes its function. For example, glucokinase is an enzyme that catalyzes the phosphorylation of glucose. Catalyzes mean it causes increased reaction using a molecule of ATP. ATP is energy molecule. The following are commonly used enzyme descriptors. So these are commonly known as enzyme names like kinase, phosphorylase, phosphatase, dehydrogenase, hydroxylase, carboxylase, mutase, synthase. First we will discuss about kinase. Kinase catalyzes transfer of phosphate group from a high energy molecule, usually ATP, to a substrate, for example phosphofructokinase. So main function of kinase is transfer of phosphate group from some high energy molecule, so like ATP, for example phosphofructokinase. Now we will discuss about phosphorylase. Phosphorylase adds inorganic phosphate onto a substrate without using ATP. So phosphorylase does not use any kind of energy or ATP. So example of phosphorylase is glycogen phosphorylase. Phosphatase, it don't add but it removes phosphate group. Phosphatase removes phosphate group from a substrate, for example fructose 1,6 bisphosphatase 1. So it's this is example of phosphatase. Dehydrogenase catalyzes oxidation reduction reactions, for example pyruvate dehydrogenase. So oxidation reduction reactions are catalyzed by dehydrogenase. Hydroxylase adds hydroxyl group onto substrates, for example tyrosine hydroxylase. Carboxylase, it transfer carboxyl group with the help of biotin, for example pyruvate carboxylase. Biotin is vitamin B7. Mutase, so mutase basically work within the same molecule. It relocates functional group within a molecule. For example vitamin B12 dependent methylmalonyl CoA mutase. This is example of this enzyme, mutase enzyme. Synthase, it catalyzes synthesis reaction without using ATP as a source of energy. So synthase does not use any kind of ATP. And phosphorylase, it works without using ATP. So these two enzymes, they do not use ATP as a source of energy. Now we will discuss about the rate determining enzyme of the metabolic processes. So first we will discuss process, then we will discuss enzyme and regulators. So these metabolic pathway is very important. It controls speed. This enzyme controls speed and it controls the whole process of reaction. First we will discuss about glycolysis. Glycolysis is formation of energy from glucose. So the main enzyme here is phosphofructokinase 1 or PFK1. It is the main enzyme in the glycolysis pathway. So activators of glycolysis are glycolysis is process of energy formation from glucose. So activators are AMP and fructose 2,6 bisphosphate. These are activator. And inhibitors, these are ATP and citrate. These two are inhibitor. inhibitors of this pathway. So how we can remember this? When there is low energy in the cell, AMP will increase, then glycolysis will also increase, and when ATP is high, so glycolysis will slow down and citrate will also decrease this process of glycolysis. Now we will discuss about gluconeogenesis. So enzyme here is fructose 1,6 bisphosphatase 1, and regulators are inhibitors are AMP and fructose 2,6 bisphosphate. These are inhibitors of this process. Gluconeogenesis is basically process of formation of new glucose molecule. So how we will remember this inhibitor? So AMP here in glycolysis process, AMP is regulator, and in gluconeogenesis, AMP is inhibitor of this process. TCA cycle. In TCA cycle, enzyme is isocitrate dehydrogenase. Activator of this process is ADP, and inhibitor are ATP and NADH. TCA cycle, the main enzyme here is isocitrate dehydrogenase. It is the main switch here. So regulators of TCA cycle are ADP and inhibitor are ATP and NADH. Glycogenesis, enzyme here is glycogen synthase, and regulator are glucose 6-phosphate, insulin and cortisol. These are regulators of glycogenesis, and inhibitor are epinephrine and glucagon. These two are inhibitor of this process. Glycogenolysis, main enzyme here is glycogen phosphorylase. Our regulators are epinephrine and glucagon and AMP. These are regulators, and inhibitors of this process are glucose 6-phosphate and insulin and ATP. These are inhibitors of this process. HMP shunt. So our main regulatory enzyme is glucose 6-phosphate dehydrogenase. So NADP positive, it is positive regulator, and NADPH, it stops this process. It is inhibitor of this reaction. De novo pyrimidine synthesis. So enzyme here is carbamyl phosphate synthetase 2, and regulators are ATP and PRP positive, and UTP is inhibitor. De novo purine synthesis, enzyme here is glutamine phosphoribosyl pyrophosphate PRPP amidotransferase. So regulators are PRP positive, AMP is inhibitor, inosine monophosphate, IMP is also inhibitor, and GMP is also inhibitor. Urea cycle, the main enzyme here is carbamyl phosphate synthetase 1, and regulator is N-acetyl glutamate. Fatty acid synthesis, the main enzyme here is acetyl CoA carboxylase. The regulators are insulin and citrate, and inhibitors are glucagon and palmitoyl CoA. These are inhibitors of this process. Fatty acid oxidation, it is the main enzyme here is carnitine acyl transferase 1, and regulator inhibitor is malonyl CoA. It is inhibitor. Ketogenesis, HMG CoA synthase. So this enzyme is main enzyme here in ketogenesis. Cholesterol synthesis, HMG CoA reductase. So insulin, thyroxine and estrogen, these are regulators of this cholesterol synthesis, and glucagon and cholesterol, they inhibit this process of cholesterol synthesis.